Good morning, good afternoon, and good evening to everyone who joins us from around the world. This is Stack's series, and we're so glad you're here.
This is Andrew and Renee, your hosts. We welcome you to the show.
Today, we are exploring the science of epigenetic clocks and the measurable pace of aging.
It's a topic that, it sounds like something straight out of a futuristic novel, doesn't it?
Oh, absolutely. But the wild part is that this technology, it's not science fiction anymore. It's already here, and it completely changes how we think about, well, the calendar.
It really does. It fundamentally shifts our perspective.
It does. When we talk about age, it's usually the most static, boring piece of information we have about ourselves. If I ask how old you are, you don't have to think about it. You show me your driver's license, we do a little subtraction, and boom, there's the number.
It's fixed. It's simple math.
Exactly. But the research we're getting into today suggests that number, the one on your birth certificate, is, well, it's kind of a deception. Or at least it's only telling half the story.
That is precisely it. We have to start with this idea that everyone, and I mean everyone, essentially has two ages.
Okay.
You've got your chronological age. That's the one tied to the calendar, the one that says when you can legally rent a car. And then you have your biological age.
The age written in your cells.
Right. The age of your tissues, in the very chemistry of your DNA. And the big aha moment that scientists have had recently, which is what we're exploring today, is that they can now measure the speed at which you're aging.
See, that's the part that really gets me. It's not just about, looking in the mirror and saying, hey, I look pretty good for 40, or feeling a bit stiff and thinking, ugh, I feel 80 today.
No, it's quantitative.
It's that scientists can look at these chemical markers on your DNA and say, okay, you've been on the planet for 40 years, but your body, it's taken 45 years worth of damage. It's like the difference between the model year of a car and the actual mileage on the odometer.
That is a fantastic analogy. How hard have you driven it? And I want to be really clear right from the start, this is not some vague guesswork.
Right. This isn't one of those online quizzes asking how many vegetables you eat. This is about measuring specific chemical tags, they're called methyl groups, that physically attach to your DNA. And they do so in patterns that change in very predictable ways over time. It's biochemistry, not magic. So we have a huge stack of sources here to get through. We've got studies from the National Health and Nutrition Examination Survey, research on muscle from the frontiers of physiology, papers on things with these kind of ominous names like GrimAge and Dunedin pace.
That's a good word for it, ominous.
But before we can even get to the clocks themselves, we have to look under the hood, right?
Yeah.
We need to understand the mechanism. We hear this word epigenetics thrown around constantly in health blogs, usually trying to sell you something.
Right, some supplement or another.
So what is it, really?
Sure.
How should we visualize the relationship between our genetics, our DNA, and this epigenetic layer?
It's the absolute fundamental question. And the best way to think of it is with an analogy. If your DNA, your genome, is the hardware of a computer, then epigenetics is the software. Or, if you like a construction analogy better, if DNA is the complete blueprint for a house, epigenetics is the contractor on the job site.
I like the blueprint one. So the DNA is the architectural plan for the entire building.
Exactly. And here's the part that people often forget. That blueprint is identical in every single room. Your eye cell contains the exact same DNA blueprint as your liver cell.
So every instruction to build a whole human is in both of those cells.
In every single one. But your eye doesn't produce bile, and your liver doesn't detect light. The question is, why?
Because the contractor, the epigenetics, is standing there telling the construction crew what to do.
Precisely. The contractor is in the eye room saying, okay, guys, ignore the kitchen plans, forget the liver plans. We are building a window right here. Turn on the lens genes, turn off the bile genes. This epigenetic layer sits literally on top of the gene. That's what epi means, on top of.
So it's a control layer, a management layer. And the main tool this contractor is using, at least for the clocks we're talking about today, is something called DNA methylation, or DNAm for short.
Correct. DNA methylation is the core mechanism.
Let's break that down, like chemically. I'm picturing that long double helix strand of DNA. What is physically happening to it?
It's a chemical tagging process. A tiny molecule called a methyl group, which is super simple, just one carbon atom bonded to 3 hydrogen atoms, comes along and attaches itself to the DNA strand.
Okay.
But it's very specific. It doesn't just stick anywhere, like, gum on your shoe. It's looking for a very specific address on the DNA highway.
An address.
Yes. It looks for a spot where a cytosine nucleotide, the C in our DNA alphabet, sits immediately next to a guanine nucleotide, the G. So it's looking for a C followed by a G. We call these CPG sites.
CPG sites, got it. So you've got these little chemical tags sticking to the DNA at these specific CG addresses. What do they actually do once they're there? Do they change the gene? Is it a mutation?
That's the crucial point. They do not change the sequence at all. The blueprint, the architectural plan remains identical. You're not erasing anything.
Okay.
Think of that methyl group like a dimmer switch on a light, or maybe a volume knob on a stereo. When a gene's promoter region, that's kind of the on button for the gene, gets heavily methylated, when it has a lot of these tags stuck to it, it essentially turns the volume way down.
It silences it.
It represses transcription. It stops that gene from being expressed, or at least reduces it significantly.
So if I'm trying to visualize this, my DNA is just covered in millions of these little dimmer switches. And as I live my life, are you saying these switches are constantly being fiddled with?
Constantly. And this is where the clock theory really comes from. For a long time, we thought these methylation patterns were either stable or just random. But when researchers started looking at them across the entire genome, scanning millions of these CPG sites, they noticed something incredible.
What was that?
The patterns aren't random at all. As we age, the placement of these methyl groups changes in a highly, highly predictable way. Some regions of your DNA gain methylation, we call that hypermethylation, and others lose it, hypomethylation.
And it happens so consistently that you can actually use it to tell time.
Yes. But to really grasp why it works as a clock, we have to bring in a concept from physics, actually. We have to talk about entropy.
Entropy,
Right.
That's usually about disorder,
Yeah.
things falling apart,
Yeah.
the universe tending towards chaos.
And it applies directly to our biology. Think of it as methylation entropy, or just noise. Let me try another analogy. Imagine a massive, pristine library.
Okay, I'm with you. In the library.
When the library first opens, so when you're young, every single book is perfectly alphabetized. The history books are in the history section. Cooking books are where they should be. The instructions are clear and crisp. The librarian, your epigenetics, knows exactly where everything is.
But then people start coming in and browsing.
Exactly. People start browsing, they pull a book off a shelf, look at it, and then put it back in the wrong spot. Someone shoves a history book into the cooking section. A page gets torn out over here. That's methylation entropy. It's not just that the dimmer switches are being turned intentionally, it's that the whole system is just getting sloppy. The signal gets noisy.
So the contractor we talked about earlier is getting a little bit senile. He's starting to lose his paperwork.
Or he's just overwhelmed by decades of work. The precise control over which genes are on and which are off starts to degrade. The pattern gets messy. And this accumulation of noise, this epigenetic drift, is what allows algorithms to predict your age. The algorithm is basically walking through the library, counting how many books are out of place, and saying, okay, based on this level of mess, this library has probably been open for about 60 years.
That's a brilliant way to put it. So it's not measuring time directly, it's measuring the accumulated chaos. And the realization that this chaos accumulates at a predictable rate.
Yeah.
That's what kicked off this whole field.
Precisely. And that brings us right to the history of these clocks. Because this science has moved at an incredible speed. We're talking about generations of technology evolving in just over a decade.
So take us back to the beginning. Who's the grandfather of this technology? Who had that aha moment for biological aging?
Well, the field really just exploded around 2013. A researcher at UCLA named Steve Horvath, he developed what we now call the pan-tissue clock. This was really generation 1.
Pan-tissue. That part sounds pretty ambitious.
It was revolutionary. I mean, before this, most researchers thought different tissues aged in completely different ways. They assumed liver aging had nothing to do with brain aging. But Horvath, he aggregated data from 51 different tissue types. We're talking blood, brain, liver, kidneys, skin, you name it. And he found a set of 353 specific CPG sites that ticked in unison across all of them.
So your liver and your brain were basically telling the same time.
Roughly, yes. He found a kind of universal metronome inside us. And the goal of this first generation Horvath's clock and another one called Hannum's clock, which focused just on blood, their goal was really specific. They wanted to guess your birth year.
Just that.
Just that. They wanted to see if they could look at a drop of your blood and accurately predict that you were born in, say, 1980.
And were they any good at it?
Incredibly good. Frighteningly accurate, actually.
Wait, hold on a second. You say frighteningly accurate like that's a bad thing? If I buy a clock I wanted to tell the right time, If I'm 40 and the test says I'm 40, isn't that a success?
In a forensic sense, absolutely yes. If you leave a drop of blood at a crime scene, the police definitely want to know that the suspect is 40 years old. And in fact, these clocks are used in forensics now. The sources mention a gene called ELVL2 that's a key marker for this.
Okay, so for crime scenes, it's great.
But for medicine, for those of us trying to improve our health and extend our health span, a clock that perfectly guesses your birthday is, well, it's kind of useless.
Because it's just confirming what the calendar already told you.
Right. Think about it. If you smoke a pack of cigarettes a day, drink heavily, never sleep, and you're under constant stress, you should be biologically older than 40. Your body's taking a beating. In that case, you want the test to fail. You want it to look at you and say, you're 55.
Okay, I get it now. We're looking for the discrepancy. We're actually hunting for the error.
The error is where all the medical insight lives. If the clock matches your birthday perfectly, it's blind to your biology. It's blind to the damage you're doing to yourself or the great things you're doing for yourself. Horvath's first clock was an amazing feat of mathematics, but it was just a really expensive way of checking your driver's license.
That makes total sense now. So the field needed a clock that could be wrong in the right way.
And that's where Generation 2 comes in, the health span clocks. This was a complete philosophical shift in how the algorithms were built. The question went from how old are you to something more like, when might you die, or how sick are you?
That is a much darker question, but I can absolutely see why it's more useful.
The big names to know here are PhenoAge and, the most famous one, GrimAge.
GrimAge. I'm going to go out on a limb and assume that's named after the Grim Reaper.
It is indeed. And honestly, the name fits. GrimAge was trained completely differently. Instead of just matching methylation patterns to chronological age, researchers trained the algorithm to predict mortality. They looked at blood markers, things like chronic inflammation levels, and even proteins associated with how many pack years someone has smoked.
So GrimAge isn't just looking at the calendar, it's looking at the wreckage. It's looking for the signs of damage.
Exactly. It analyzes DNA methylation as a proxy, as a surrogate for these plasma proteins that indicate stress and disease. It's looking for the smoke that tells you there's a fire. And why this matters is, if GrimAge says you're 60, but your birth certificate says you're 50, that's what we call age acceleration.
You're aging faster than you should be.
Right. And if your GrimAge is 10 years older than your chronological age, you are at a significantly higher risk for cardiovascular disease, for cancer, for all-cause mortality. It's a much, much better predictor of your remaining lifespan than your birthday is. It finally catches the impact of the smoking, the stress, the bad diet.
So to go back to the car analogy, the first clocks told you the model year. GrimAge tells you the total mileage on the odometer.
Perfect analogy. It tells you this car has 100,000 miles of wear and tear, and it doesn't care if it's a 2020 model or a 2010 model.
But even GrimAge is kind of a snapshot, isn't it? It's telling you how much damage you've accumulated up to this very moment. It's looking backward.
That's correct. And that is a limitation. If you decide to change your lifestyle today, you start eating well, exercising, sleeping, the odometer on your car doesn't roll back immediately. It takes a long, long time to change the total mileage of the car.
So we need a speedometer. We need to know how fast we're driving right now.
Exactly. And that leads us to Generation 3, the speedometers. And to understand this generation, we have to talk about a place called Dunedin.
Dunedin Pace and Dunedin Pom. I saw these names in our sources.
Yeah.
This comes from that absolutely massive study in New Zealand, right?
Yes. And we have to just pause for a second and appreciate the Dunedin study. It is, and I'm not exaggerating, it is arguably the most impressive longitudinal study in human history.
Wow.
It started in Dunedin, New Zealand. They took about 1,000 babies, all born between 1972 and 1973.
Just a random group of babies from that town.
Basically a representative sample of the population. And normally in these really long-term studies, people drop out, they move away, they lose interest, life happens. But the retention rate in the Dunedin study is legendary. It's something like 95% nearly 5 decades later.
That's unheard of. So they had the same 1,000 people checking in every few years for their entire lives.
Every few years, they bring them into the clinic and they measure everything. I mean, lung function, dental health, retinal imaging, renal function, liver enzymes. 18 distinct biomarkers of organ system integrity. And because they did this at age 26 and then again at 32 and 38 and 45, they didn't just get a snapshot. They got a movie.
I see where you're going with this.
Yeah.
The other clocks, GrimAge, Horvath, Those are snapshots. They compare you to a huge database of other people.
Correct. But the Dunyan team could look at the change in those 18 biomarkers within the same person over time. They could calculate a velocity, a rate of change.
So instead of answering, how old are you, it answers the question, how fast are you driving toward the cliff right now?
That's exactly it. It gives you a rate. For example, a Dunyan pace rate of 1.2 means that for every 1 calendar year that passes, your body is aging 1.2 biological years. You're in the fast lane. You're putting 14 months of wear and tear on your body every 12 months.
Or, on the flip side, if you're really healthy, maybe you're aging at 0.8 years for every chronological year. You're effectively aging in slow motion.
Precisely. And this distinction is so crucial for actually testing interventions. Because if I change my diet today, the speedometer, the Dunyan pace, might show a change in just a few months. It reacts to your current conditions. Whereas the odometer, GrimAge, might take years to show a statistically significant difference.
That is the perfect way to explain it. If I slam on the brakes in my car, the speedometer drops to 0 instantly, even if the total mileage on the car stays the same for a while.
Exactly. And that makes Dunyan pace the gold standard right now for clinical trials and for anyone doing personal experiments to see what works for them.
So is that it? Is there anything coming after this, or have we kind of reached the peak of this technology?
Now, science never sleeps. We're already entering what you could call Generation 4. People are now using neural networks and AI to find nonlinear patterns that the simpler models might have missed. And we're developing what are called causal clocks.
Causal, as in trying to figure out which of these methylation sites actually cause the aging, rather than just the ones that happen to correlate with it.
Yes. Trying to separate the passengers from the driver. Just because a gene gets methylated as you get older doesn't mean it's causing the aging. It could just be a bystander, a consequence of something else. Causal clocks try to find the root drivers of the process.
And what about specializing? Are we seeing clocks for specific organs?
We are. There's a cortical clock that's specifically designed for brain aging and predicting Alzheimer's risk because, as we kind of hinted at earlier, the idea that your whole body is one single age is probably too simple. Your brain might be a different biological age than your liver.
That is such a trippy thought. My brain could be 40, but my knees could be 70.
It's entirely possible biologically. I mean, different tissues face different stressors, right? Your skin is exposed to UV light. Your liver has to process toxins. Your heart is under constant mechanical strain. It makes sense that they could age at different rates depending on your genetics and how you treat them.
So we've established that this clock is ticking, and it's ticking faster for some people than others, based on all these factors. But there's a huge data point in our sources that we haven't touched on yet. And it has to do with the fact that, well, women and men don't age in the same way.
The famous gender gap in longevity.
Exactly.
Right.
Because logically, you think, okay, women live longer than men on average, so their clocks must just tick slower. But the data on major reproductive events like menopause and pregnancy, it suggests something much more volatile is happening.
It's a fascinating paradox. Women do live longer, they have greater longevity, but they also often report higher morbidity, more sickness, more chronic conditions, more frailty in those later years. And the interaction between their hormones and these epigenetic clocks is just massive.
Let's start with one of the biggest transitions, menopause. The PhenoAge study, that's a big national health survey in the U.S., had some really specific findings on this, didn't it?
It did. They used the GrimAge clock, the odometer, to look at thousands of women and their menopause timing. And the finding was quite stark. A later onset of menopause is associated with a lower epigenetic age.
So if you go through menopause later in life, your biological clock is effectively younger.
Correct. And the flip side of that is that early menopause isn't just a reproductive event. It's not just about fertility ending. It signals a systemic acceleration of aging risks across the body.
And the mechanism there is what we think it is, estrogen.
It seems to be tied directly to estrogen. Estrogen is a powerhouse hormone. It's somewhat protective for the cardiovascular system for bone density, and it appears for the epigenome itself.
It's like a protective shield.
In a way, yes. So when you lose that shield during the menopausal transition, and that's not just a single day, it can be a process lasting anywhere from 2 to 8 years, the loss of that hormonal protection seems to press the accelerator on the aging clock. The study suggests this transition period is a critical window where biological aging speeds up.
What about the other end of the spectrum? Puberty? Menarche?
That's a bit more complex. Menarche is the first occurrence of menstruation. And some of the smaller, earlier studies suggested that an early menarche, so starting your period at very young age, was linked to accelerated aging later in life. The theory was sort of the clock started ticking sooner.
But the bigger data didn't back that up.
No. The large NHENE study that we're referencing actually found no significant link between the age of puberty and a person's epigenetic age later in life.
That's really interesting. So puberty kind of sets the stage, but it doesn't necessarily set the speed of the clock.
It appears so. You can think of puberty as an epigenetic gatekeeper.
Wow.
The massive hormonal reorganization. You have GnRH, LH, FSH all coming online. It definitely imprints the epigenome. It transitions the body from a child state to an adult state. But it seems to be the menopause transition that is the more active accelerator of the clock in later life.
Okay, now for the finding that really truly blew my mind, pregnancy. The findings out of Yale.
This is just fascinating science. We've always known intuitively that pregnancy is hard on the body. I mean, anyone who has been pregnant or lived with someone who is pregnant knows this. But the epigenetic clocks quantify it in a completely new way.
How so?
Pregnancy is biologically and metabolically incredibly expensive. It's a massive stress test for the body. And the clocks reflect this. During pregnancy, a woman's epigenetic age accelerates. She literally ages faster.
You're kidding. So if you measure a woman's biological age at, say, 3 months pregnant, and then again at 8 months pregnant, she's aged more than just those 5 calendar months.
Biologically, yes. The system is under enormous strain. Her cells are working overtime to build a whole new human. But, and this is the incredible part, this aging appears to be reversible postpartum.
It reverses. It actually goes back.
It's what they call a stress recovery model. After the baby is born and the mother's body has time to recover, her biological age can drop back down, often all the way to her pre-pregnancy baseline.
That is wild. It suggests that our biological age isn't a one-way street. It's plastic, it's elastic, it can stretch out and then snap back.
That is the key insight from this line of research. Aging isn't just a linear slide downwards. It fluctuates with stress. It's dynamic.
Does it always snap back all the way? The source mentioned parity, which is the number of children a woman has had.
That is the big open question. High parity, having many children, especially close together, might be linked to different aging outcomes. It likely depends on the recovery period. Think of it like a rubber band. You stretch it, it snaps back. But if you stretch it again and again and again without letting it fully rest, it might lose some of its elasticity. It might get a little loose over time.
So the recovery period is just as important, if not more important, than the stress event itself.
Crucially so. And this concept applies to everyone, not just in pregnancy. A major surgery, a severe infection like COVID-19, a period of extreme psychological stress. These all seem to temporarily accelerate biological age. But when we recover, the age can recede. Health is about resilience.
Speaking of structure and stress, let's talk about the actual hardware of the body. Part 4, the muscle-bone connection. I feel like we so often think of muscle as just aesthetic. You know, looking good at the beach or being able to lift a heavy box. But the expert view in these papers is that muscle is an organ of longevity.
We absolutely have to reframe how we view muscle. It is not just slabs of meat for moving your skeleton around. It is a metabolic engine. It is an endocrine organ.
An endocrine organ, you mean like the thyroid or the pancreas?
Yes. Active muscle releases these signaling molecules called myokines. These are chemical messengers that travel through your blood and talk to your brain, your immune system, your liver, your bones. Muscle is also the largest site for glucose disposal in your entire body. If you want to manage your blood sugar, muscle is your best friend. It's like a giant sponge.
The sources bring up 2 terms for muscle loss: sarcopenia and dynapenia. I think I've heard of sarcopenia, but can you help distinguish them?
Sure. Sarcopenia is the age-related loss of muscle mass. The muscle physically shrinks, you have less of it. Dynapenia is the age-related loss of muscle strength. And here's the kicker. They don't always happen at the same rate.
So you could have muscle that looks okay size-wise, but it's actually really weak.
Exactly. Think of it like a cheap, fatty steak versus a lean, high-quality one. As we age, if we aren't careful, our muscle tissue can get infiltrated with fat. It gets marbled. This is called intramuscular adipose tissue. So your arm might look the same size in a shirt, but the actual contractile tissue, the engine part of the muscle, is being replaced by metabolically dysfunctional fat. That's dynapenia. the quality of the muscle degrades.
And this just gets worse and worse as we get older.
It can become a perfect storm of aging. After about age 60, lean mass can decline pretty substantially. You have inactivity combined with something called anabolic resistance.
Anabolic resistance. That sounds like a resistance to steroids, but I'm guessing it means something else in this context.
It does. It means your muscle stops listening to growth signals. When you're 20, you eat a steak or a protein shake, you lift a weight, and your body gets a loud and clear signal. Great, let's build muscle. When you're 70, you do the exact same thing, but the muscle acts like it didn't hear the message. It becomes resistant or deaf to the protein signal.
That is so frustrating. So you have to work twice as hard to get the same result.
You do, but you must do it because it's not just about the muscles themselves. They are in constant communication with your bones.
This is that biomechanical coupling idea.
Yes, muscles and bones are physically and chemically linked. Physically, your muscles attach to your bones via tendons. When you contract a muscle, you are literally pulling on the bone. That mechanical stress, that bending and pulling, is a signal to your bone cells. It tells them, hey, we're being used, we're under load, we need to get stronger.
Use it or lose it.
Literally. Bones are incredibly smart tissue. If they aren't being pulled on in stress, they start to dissolve. Your body sheds the calcium because it's metabolically expensive to maintain a strong skeleton you aren't using. There was a great study on postmenopausal women showing that those with a higher appendicular skeletal muscle mass index, basically more muscle on their arms and legs, had significantly better bone microarchitecture. Their bones were just structurally sounder.
So strong muscles pull on bones,
Yeah.
which makes for strong bones. It's a virtuous cycle.
It is. And if you're inactive, you lose both. This brings up the important concept of secondary aging.
What's that?
Primary aging is the inevitable stuff, right? Time passes, entropy happens, the library gets a little messy no matter what. Secondary aging is the decline that is caused by our lifestyle, and particularly inactivity. Sarcopenia and osteoporosis are largely driven by secondary aging. And that's good news because it means they are potentially reversible, or at least preventable. You don't have to become frail just because you get old.
That's a perfect segue to our final major section, lifestyle and environmental influences. Because if this clock is measuring speed and if muscle is this huge metabolic engine, we all want to know, where's the brake pedal?
And just as importantly, what's pushing down on the accelerator?
The sources distinguish between intrinsic and extrinsic aging. Can you break that down for us?
Sure. Intrinsic aging, or IEAA, is the cell internal stuff. It's the developmental clock ticking. It's harder to change. It's like the battery slowly draining on your phone over years of use. Extrinsic aging, or EEAA, is largely about the immune system and inflammation. And this is the part that is highly responsive to lifestyle. This is where your choices matter most.
So let's talk about the big one, diet. Are there really anti-aging foods according to the epigenetic clock? Or is it just the boring advice to eat your veggies?
Well, it is eat your veggies, but now we actually know why on a mechanistic level. The data points to protective effects from fish, poultry, fruits, and vegetables. Specifically, foods high in things like carotenoids and nutrients that support the body's methylation processes. But there's this really cool concept of methyl adaptogens.
Methyl adaptogens, that sounds very fancy.
It refers to compounds in foods that can actually influence the DNA methylation machinery itself. For example, sulforaphane, which you get from broccoli and other cruciferous vegetables, or EGCG from green tea, or curcumin from turmeric. These aren't just vitamins, they interact with the enzymes, the contractors, that are placing the methyl tags on your DNA. They help the contractor stay focused and do his job properly.
So you're literally feeding the contractor.
Exactly. You're giving him the right tools to keep the blueprints clean and the library organized.
And what about the flip side? What foods are pushing the accelerator?
The biggest one, by far, is obesity and high BMI. They are strongly linked to accelerated epigenetic aging. But it's important to understand why. It's not just about carrying extra weight. It's about inflammation. Adipose tissue, especially visceral fat, that's the hard, dangerous fat around your organs, isn't just a passive storage depot for energy, it's a biologically active endocrine organ, and it pumps out inflammatory cytokines.
So excess fat is basically an organ that's constantly shouting panic at the rest of your body.
That's a great way to put it. It creates a state of chronic, low-grade inflammation, and that inflammation forces cells to divide faster to repair damage, which accelerates the ticking of the clock.
What about movement? We talked about building muscle, but what about just not sitting all day?
This is the sedentary pandemic, and the mechanism here is actually pretty scary. The sources describe inactivity not just as the absence of exercise, but as an active signal of decay being sent to your body.
Wait, unpack that for me. I always thought sitting was just sort of neutral, doing nothing.
It is not neutral. If you go for a run for 30 minutes, you create a positive metabolic signal. But if you then sit for 10 hours straight, the sit signal, the metabolic stagnation, the blood pooling, the lack of muscle contraction, imposes a negative physiological state that can persist longer and be more powerful than the positive signal from the exercise.
So the sit signal creates a louder noise in the system than the run signal does.
It can absolutely drown it out.
Right.
Inactivity sends a signal to your body that basically says, we aren't moving, so we don't need to repair things. Let's down-regulate the repair systems. Let's conserve energy. Let's store fat. It's a powerful accelerator of that extrinsic inflammatory aging clock.
That is a very stark warning. We really do need to break up our sitting time.
Absolutely. Even just standing up or doing a few squats every hour helps break that signal.
Okay, let's talk about sleep. I feel like everyone knows they should sleep more. But the link to the epigenetic clock is really specific, isn't it?
It is. Specifically, it's about sleep apnea. This is a massive, massive accelerator.
And why is that? Is it just about being tired all the time?
No, it's about hypoxia, a lack of oxygen. When you have sleep apnea, you stop breathing, sometimes hundreds of times during the night. Your blood oxygen levels plummet. Then you gasp for air, and they spike back up. This constant fluctuation causes enormous oxidative stress. It inflames the entire system.
Wow.
Severe untreated sleep apnea can add hundreds of days, sometimes even years, to your biological age compared to your chronological age.
That is terrifying. But the good news is it's treatable.
Highly treatable with CPAP machines, dental devices, lifestyle changes, and studies are now showing that treating the apnea can slow the clock back down.
One more factor in this section that I found really compelling. Socioeconomic status, or SES, and this is where the science gets really sociological.
It's unavoidable. The GrimAge clock in particular is very sensitive to social disadvantage. And we're seeing that childhood adversity, things like trauma, poverty, low socioeconomic status early in life, it leaves epigenetic scars.
So you're saying poverty literally gets written into your DNA?
Into your epigenome, yes. It can accelerate your biological aging decades later. Even if you're wealthy and successful at 50, the stress of being poor at age 5 might still be ticking away in your methylation patterns. We see that higher education correlates with slower aging, and that's likely a proxy for better lifestyle, less environmental stress, better access to nutrition. But it shows that inequality is a biological issue, not just a social one.
That is profound. It means our personal history is encoded in us in a way we never really understood before.
It absolutely is. But remember the lesson from the pregnancy study. It's plastic. It can be modified.
Which brings us right to the future. Reversibility. Can we actually turn the clock back?
This is the absolute frontier. In the lab, in Petri dishes, scientists can use things called Yamanaka factors to completely reset a cell all the way back to 0 to an embryonic stem cell state. That is the extreme version.
That sounds like turning the butterfly back into a caterpillar.
It is. And we don't want to do that to your whole body. That would be very bad. You'd basically turn into a blob of jelly. But the idea of partial reprogramming, using these factors to turn the clock back just a little bit, to make a cell more youthful without it losing its identity, that's the holy grail of aging research.
But for now, for us listeners, we don't have access to Yamanaka factors. We have lifestyle.
Correct. For us, the recovery concept is our version of reprogramming. Biological age increases with stress surgery, COVID, pregnancy, but upon recovery, it can return to baseline. The goal is to build resilience, to be able to bounce back from insults faster and more completely.
And it shifts the entire paradigm of medicine from treating the sick to maintaining the young.
That's the dream. If we can slow the rate of aging, if we can lower that Dundon-Pace number from a 1.2 to a 0.8, we delay the onset of all age-related diseases at once. Cancer, heart disease, Alzheimer's, diabetes. They all strike later if the clock ticks slower. The goal is to add more healthy years to your life, to improve health span.
There's a big forensic and ethical side to this as well, isn't there?
We touched on it. But imagine an insurance company requiring a GrimAge test for a life insurance policy. They wouldn't care when you were born. They would have a pretty good idea of your predicted date of death based on your methylation.
Sorry, your premium is triple because your GrimAge is 75 even though you're only 50.
It raises massive privacy implications. It's the most personal biological data you have. But it also empowers the individual. If you know your rate of aging is fast, you can intervene. You can get a test, make some changes, and then test again 6 months later to see if it worked. It provides direct feedback.
So at the end of all this, what does this mean for the listener right now, today?
It means you are not a slave to the calendar. Your birth certificate is a historical document, not a biological diagnosis. You have a steering wheel. You have a brake pedal. Through your choices in diet, movement, sleep, and stress management, you are actively participating. You are editing the software of your own biology every single day.
That is an incredibly powerful thought. You are the editor of your own genetic story.
To a very significant degree, yes.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support.
And to our listeners, thank you for being part of this journey with us. If you enjoyed today's episode, be sure to subscribe and follow Stack Studios for more informative content, and support us in Patreon.
Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.